Very much like lysosomes and vesicles, there are other organelles whose structure resembles a bubble or a small sac of membrane. First of all, vacuoles fit this bubble of membrane category, but depending on their use, vacuoles can vary widely in size. In some plant cells, they occupy up to 90% of the space inside the cell. I think of vacuoles as the storage closet of the cell. They can store water or another aqueous solution. So sometimes those aqueous solutions include pigments, which could attract pollinators, thus aiding in reproduction, or sometimes they're toxins, which to a plant cell is going to be very important to help deter herbivores and predation. Sometimes they contain enzymes, which I know makes them very similar to lysosomes, but we're making the distinction that lysosomes are in animal cells and vacuoles, while they can be found based on some books in animal cells, we're talking about them primarily in plant cells. And as I said before, vacuoles are often for storage. Storing water does more for the plant than simply preventing dehydration. As the vacuole grows, it puts pressure on another organelle that's not found in animals, and that's the plant cell wall. So much like air pressure can make the walls of a balloon stiff, the water pressure makes the plant cell stiff and sturdy. And so that's going to maintain something called turgor pressure, and it's the main reason that plants wilt when they need water. And so if you look here, here's an example of a plant cell that has a full vacuole there in the center, and so it's putting pressure on the orange cell walls. And this would be an example of a wilting but still living plant. And so the cell walls can collapse down a little bit without that water pressure or turgor pressure there. And then this would be an example of what could happen when your plant cells start to die and turn brown. If the vacuole gets too small, the cell membrane will actually peel away from the cell wall, causing something called plasmolysis. And so that's very bad for the plant. Moving on, peroxisomes do what their name implies. They collect peroxides like H2O2 and break them down using special enzymes. Peroxides are unavoidable. Even breathing oxygen can cause them to form as a byproduct of metabolism. So almost all eukaryotes, including animal and plant cells, need peroxisomes sometimes. Speaking of metabolism, let's take a look at the two famous energy-converting organelles, and that would be your mitochondria and your chloroplasts. All eukaryotes have mitochondria since they are the main source or main site for cellular respiration. And so glucose is what we're going to start out with when we're talking about cellular respiration. And when glucose is taken into the cell, it is broken down in the cytoplasm and cleaved in half to form two molecules called pyruvate. The pyruvate enters the mitochondrion, which is just the singular form of mitochondria, and is broken down by a series of energy-capturing reactions, finally yielding carbon dioxide and water. So we've got many arrows there symbolizing the many reactions. The energy is converted into ATP. So that energy that's released is captured and turned into energy stored in the bonds of ATP. This is all possible because of the unique shape of the mitochondria. They have two membranes. The outer membrane is smooth and the inner is folded. Yet again, those folds increase the surface area of the membrane. The inner membrane is full of proteins that conduct the many steps of cellular respiration, which we'll talk much more about when we discuss cellular respiration in detail. The space inside that inner membrane is called the mitochondrial matrix, and that matrix also includes ribosomes and DNA specific and unique to the mitochondria. So, yes, this organelle has its own DNA. It can make some of the proteins it needs for cellular respiration, but other proteins needed for cellular respiration it has to get from genes stored inside the nucleus. So it can't do it all on its own. So the mitochondria's purpose is very clear. It's to turn organic molecules into a form of energy the cell can use, that's ATP. So it should not surprise you that in cells with higher energy needs, you find cells with much more mitochondria per unit of volume than you would in cells with low energy needs. Now for the chloroplast. The chloroplast is found only in plants. It can also be found in eukaryotes that have algae living within them in a symbiotic relationship, so this includes corals and lichens. Chloroplasts are green because of the pigment chlorophyll. Chlorophyll helps to capture photon energy, and because photosynthesis is the primary source of all Earth's biomass, those organelles and their chlorophyll are creating most of the food source for the entire planet. Chloroplasts also have their own DNA and ribosomes, and they also have an inner membrane. In fact, they have an outer membrane, an inner membrane, and a set of membrane compartments suspended in a fluid that's called the stroma. Each little compartment is called a thylakoid, and then a stack of thylakoids, so these disc-shaped membranes, is called a granum, and plural is grana, so here are the grana. And those are connected by membranes to each other. And then there again is the stroma, the liquid that they are suspended within. All along the thylakoid membrane, if we were to zoom in really close, you could see the proteins that are responsible for photosynthesis. And here's the equation for photosynthesis. So at this point, hopefully you are seeing some definite similarities between mitochondria and chloroplasts, including their energy conversion processes. They're mirror images of each other. So if you're not seeing a lot of similarities yet, go back, watch the mitochondria bit again, look at the structure, look at and listen to details about them. You should start to see some of those comparisons emerge. And that brings us to some pictures of the chloroplast. So you can see a little toy down there in the bottom corner showing the thylakoids. And I love this because it's actually showing how the thylakoids are really not individual discs. They're connected to each other in some parts. And then you can see that transmission electron micrograph, so seeing what a chloroplast really looks like through a microscope. And finally, endosymbiotic theory. So when we're talking about the size of mitochondria and chloroplasts, they're very similar to prokaryotes. And so a lot of the similarities between mitochondria and chloroplasts have led to what we now call the endosymbiotic theory, this idea that mitochondria and chloroplasts are the distant relatives of prokaryotes. So they also have two membranes, so that makes us think that they might have been ingested by an early prokaryote. One prokaryote eating another was very common, but in this case they might have stuck around and not been consumed. They have DNA and ribosomes just like an independently living creature, and they can divide all on their own. And so this would have allowed them in the early days to coexist with their prokaryote host. They would have continued to divide and replicate on their own like an independent cell, and so as their host cell divided, they would have stayed within their host cell. And so all of these things have led us to form this statement that kind of summarizes endosymbiotic theory, which endo for in, symbiotic for a symbiotic relationship. And so we think that early eukaryotes were actually the result of a symbiotic relationship between one prokaryote living inside of another. We have been further developing this theory with modern DNA sequencing. So we've compared the DNA in these organelles to similar prokaryotes with similar functions, and they're not exactly the same, of course, but they're very similar. And while neither organelle can survive on its own anymore, it is likely that billions of years ago, their ancestors might have been independently living creatures. So I just want to wrap this up by just, again, thinking of the biochemical similarities between prokaryotes and eukaryotes. They share a lot in common. We have DNA as our genetic material, and we not only use amino acids to build our proteins, but we use primarily the same isomer of those 20 amino acids. So there's a lot of biochemical evidence that also favors this conclusion of endosymbiotic theory. So that's it for this part of the organelles lesson. The next screencast is going to include the cytoskeleton. So check that out next.